Hello students, so we are going to talk about chapter 14, Mendel and the gene idea. So we want to know, who is Gregor Mendel? Gregor Mendel is called the father of genetics. And as we can see here from right, 1 to 3, that Gregor Mendel, he is the one that is holding the sprig of fuchsia with his fellow monks. He is called the father of genetics. He worked on the garden pea plant to study all these 7 pairs of contrasting traits. And this is in the slide, Mendel's 7 pairs of contrasting traits. Now, he worked on this garden pea plant, and the pea plant in botanical or scientific terms is called Pisum sativum. So what are the 7 pairs of contrasting traits that Mendel studied? Was he looked at flower color, seed shape, pod color, pod shape, flower position, and the length of the stem, or in other words, the height of the plant. So he looked at dominant traits versus recessive traits. And a dominant trait is a trait that is expressed in a generation. Opposite of dominant trait is a recessive trait, a trait that lies hidden. It is masked, M-A-S-K-E-D, in a generation. In simple words, it does not show up in a generation. So purple flowers are always dominant over white flowers. When he looked at seed color, he compared yellow seeds versus green. So yellow seeds were dominant over green seeds. When he looked at the shape of seeds, which are pea seeds, round seeds were dominant over wrinkled. Peas come in a pod. So green pod color was dominant over yellow pod color. When he looked at pod shape, inflated pods that were inflated were dominant, and constricted were, which means narrow, were recessive. Flower position, axial flowers are dominant over terminal. Axial comes from the word axis. So if you look at the stem, the branch, and the leaf, in between this angle, axis, angle, the flower is located between the stem and the leaf, the branch which consists of the leaf. Terminal, look at the position of the flowers. They are at the tip of the stem. So this is terminal, which is recessive. And the last but not the least is plant height or stem length. Tall plants versus is dominant over dwarf plants. Now, what I want you to write next to each of these dominant traits and recessive traits are the letters. Dominant trait, as I explained, is a trait that is expressed in a generation. I give you a clue sentence. It is always indicated in capital letters. So you are looking at the first letter of the spelling of the word, which is purple. And you're going to put capital P, it's a pair, into parentheses, versus white, which is lowercase p, lowercase p. So this is important to remember, that you use the same letter of the dominant, but it is written in lowercase letters. Yellow would be capital Y, lowercase y. Round begins with the letter R, capital R into parentheses, lowercase r, spelling of the same. You're writing the same letter in lowercase form. Now pod color, capital G's, lowercase g's for yellow. Inflated and axial, we won't write any genotypes. Tall, capital T, lowercase t, lowercase t. So now, what I'm going to show you is, we're going to flip these and we are going to go into all the definitions, 7 definitions and concepts of genetics. So we talked what a dominant allele is, or a dominant trait, a trait that is expressed in a generation. It is always written in capital letters. For example, purple flowers, capital P, capital P. A recessive trait, a trait that is not expressed. It lies hidden or it is masked. Second point, it is always written in lowercase letters. So opposite of purple was white flowers. So white would be lowercase p, lowercase p. Then we're going to go down and we're going to look at this term here, which is homozygous. Homo comes from the word same. Homo means same. So the definition I'm giving you is, when the alleles for a given gene is the same, then the condition is said to be homozygous. I repeat, homo comes from the word same. So when the alleles for a given gene is the same, the condition is said to be homozygous. Therefore, let's keep the same example, purple flowers. How do we write purple flowers homozygous condition? Capital P, capital P. How would I write the contrasting trait, white flowers? Lowercase p, lowercase p. Opposite of homozygous is heterozygous. Hetero comes from the word different. Therefore, when the alleles for a given gene is different. So different is your keyword. Let's keep the same example, purple flowers. So now we're going to write one capital P and one lowercase p. That condition is said to be heterozygous purple. Homozygous purple, capital P, capital P. Hetero means different, so capital P, lowercase p, like here. Capital P, homozygous purple, capital P, lowercase p, heterozygous purple. Let's go to the term called phenotype. See the clue sentence I'm giving you. P, P. What does the second P refer to? The physical or the outward appearance of an individual is referred to its phenotype. So if I'm talking about the phenotype of purple flowers, the second clue sentence I give you, it is the spelling of the word. And spelling of the word means, how do we spell purple? P-U-R-P-L-E, purple. How do we spell white? W-H-I-T-E. So that's the phenotype of that trait. Opposite of phenotype is genotype. Geno, genetic composition. So genotype is the genetic makeup or the genetic composition of an individual. Therefore, what's the genotype? How do we express the genotype? It's always expressed in the form of letters. So capital P is the genotype for purple flowers. Capital P, lowercase p is the genotype for purple. And I could say lowercase p is the genotype for white flowers. So you can have homozygous purple, heterozygous purple, and you only can have homozygous recessive white flowers. So just to reiterate, I've given you the definition of dominant, recessive, homozygous, heterozygous, genotype, phenotype. And now 2 more definitions. What we're going to talk about is a ratio. So we're going to look at what is a monohybrid cross and a dihybrid cross. So let me go back here to explaining what a monohybrid cross is. So monohybrid comes from the word, mono comes from the word one. So a monohybrid cross is a cross in which one pair of contrasting traits is studied. So a classic example would be purple flowers versus white flowers. Number two, the ratio in a monohybrid cross is always 3:1. Opposite of a monohybrid cross is what we call a dihybrid cross. So dihybrid is when two pairs of contrasting traits are studied. For example, yellow, the color of the pea versus the texture, round peas, green versus wrinkled. So two pairs, 1, 2, 3, 4. 4 times 4, you'll get 16. So the ratio in a dihybrid cross is 9:3:3:1. So slide 20 is explaining the dihybrid cross, which I will be explaining to you later. I will also work out for you in the class a dihybrid cross in the lab, as well as try to work out monohybrid crosses in the lab when we do Mendelian genetics. So now let's get back to the slide. So what did Mendel do? He worked on these test crosses. So he took purple flowers and he mated them with white flowers. We know purple are dominant and white flowers with the recessive trait. But keep in mind, in flowers have both the male and female sexes present. So how did he work on these genetic experiments? He took the male part of the flower called the stamen. And what is the stamen comprised of? It is made up of this whole structure. So the anther, which contains the pollen, the filament, and that makes up the whole stamen. So this is the whole stamen as they show. This whole region is the female sex organ of a flower, which is called the carpel or the pistil. So it consists of the sticky stigma, the style, and the ovary, which contains ovules. Ovules are the seeds. So when you eat fruits like peaches, nectarines, plums, we are eating the female sex organ called the ovaries, and the seeds are the ovules. So he conducted self-pollination experiments, wherein you can take the paintbrush, you can dust the pollen from the anther, which is the male stamen, and touch it to the pistil or the carpel, which is the female, and pollination will take place. So transfer, what is pollination? Transfer of this anther, or of the pollen grains from the anther to the sticky stigma is called pollination. Self-pollination. It's occurring on the same plant. If you take it, dust the pollen from one plant and transfer it to another plant, then it's called cross-pollination. And so let's see what Mendel did. So whom do you cross? You cross the parents. The P1 generation of the parents. So it would be capital P versus capital P versus lowercase p. So I want to go back to here. Let's go to this cross. So you take capital P, homozygous dominant purple, versus homozygous recessive white. So you underline one from each pair. So you'll get capital P, lowercase p. And now, so capital P, lowercase p, and then capital P, lowercase p, which will be the heterozygous. So the F1 generation, F1 stands for one, first, F, filial, F-I-L-I-A-L, which means sons and daughters in Latin, generation. So you get the first filial generation, which is capital P, lowercase p, heterozygous purple. You cross it with another heterozygous purple. And now we make what we call the Punnett square. So what is this Punnett square? This Punnett square is a checkerboard, C-H-E-C-K-E-R. It's a checkerboard that is used to show the offspring. These are all the offspring obtained in the cross, the test cross that we are carrying out. Now, on one side of the checkerboard, you put either the male gamete. A gamete is a sex cell, which is the sperm cell. And on the other side of the checkerboard, you put the female gamete, which is the egg cell. So now how do we get the offspring? So always capital letters come first, the dominant trait. So capital P goes here. You're matching. Capital P, lowercase p goes here. Now you're finished with this, go to the next box. Capital P, lowercase p goes here. Capital P, lowercase p comes here. Lowercase p goes here. So now how do we prove this 3:1 ratio? So let's underline capital P. So you get one capital P, one capital P, one capital P. So there are 1, 2, 3. But you have one homozygous, so you have 3 purples with 1 white flower. But there is one homozygous dominant purple with 2 heterozygous dominant purple and 1 heterozygous white recessive. So always the 3:1 ratio is 1 homozygous dominant with 2 heterozygous dominant purple, 1 homozygous white. So it's a 1:2:1 ratio. So now, I like you to use the 7 pairs of contrasting traits and keep practicing the monohybrid crosses. I will teach you how to perform a dihybrid cross, which is the 9:3:3:1 ratio. Now, when you look at this monohybrid cross here, let's look at this cross. 3:1 means 3 dominant, 3 out of 4. And that's why you took a pair. 2 times 2 is 4. So you know you have to get 4 offspring. Whereas when you took 2 pairs of contrasting traits, like I'm going to show you here, it was 1, 2, 3, 4 letters. 4 times 4 is 16. So you're drawing a 16 checkerboard. And so I'm going to explain this exclusively in the class when we perform genetics. So now let's go to what alleles were. I gave this under structure for chromosome and mitosis. There are 2 or more alternate forms of a gene or versions of a gene. And so here they're just trying to point out the alleles for purple flowers. You know, in the nucleic acids on DNA, we looked at cytosine, thymine, adenine, thymine, cytosine. And so if this one is changed, the color can change. And that's what they're trying to show you, or the absence of an enzyme will result in insufficient pigmentation. Now, Mendel put forth certain laws of inheritance. One law was called the law of segregation. Second law is called law of independent assortment. So what is the law of segregation? In English, segregation, to segregate is to separate. So law of segregation states that the two alleles for a given characteristic or trait will separate out during gamete formation and end up in different gametes. In other words, to put it into perspective, the law of segregation is stating that every sexa, every gamete will receive one of the copies of the two alleles for a given trait. So thus the egg cell, which is the female gamete, will receive one of the two alleles. The sperm cell will receive the other allele from that pair of alleles. And so law of segregation always accounts for the 3:1 ratio. Now, I explained to you what that Punnett square was, the checkerboard. So let's go now to, we've conducted the test graphs, the second law of inheritance, the law of independent assortment. What is the law? Very important, students. These laws keep in mind and remember. Law of independent assortment states that each pair of alleles is going to separate out independent of the other pair of alleles during gamete formation. So where does this law apply to? This law is applying not to the homologous chromosomes that we saw in the karyotype and structure for chromosome in mitosis. It refers to the genes on different non-homologous chromosomes. And what they say, that those genes that are close together are inherited together. Those genes, so if a gene is here, gene is here, gene is here on the chromosome, these genes will be inherited together. Genes that are further apart on the chromosome will be inherited together. So it applies only to genes on different non-homologous chromosomes or those far apart on the same chromosome. Genes located near each other on the same chromosome tend to be inherited together. This is a key factor that you must remember. This is the law of, this is the dihybrid ratio that we will perform on the board in genetics. Now we come to other degrees of dominance. You have law of complete dominance. What is the law of complete dominance states? At one trait is completely dominant over the other. But look at the jargon, scientific jargon of terms. We say when phenotypes of the heterozygote and dominant homozygote are identical. We don't understand anything. But to put into context, the law of complete dominance states that one trait is completely dominant over the other. For example, purple flowers are completely dominant over white flowers. Let's go to the second law of dominance. It is the law of incomplete dominance. Incomplete means one trait cannot be completely dominant over the other trait. Therefore, what are we saying in the law of incomplete dominance? We are saying that one trait is not completely dominant over the other, but the heterozygote lies in between that of the dominant trait and the recessive trait. This is the classic example of incomplete dominance. This red snapdragons, this is a particular flower. Snapdragons are not insects. This is the beautiful red snapdragon flower. When red snapdragons are crossed with white snapdragons, how do we denote the alleles for red snapdragons? It's CRCR. White CW. We would never write that for a dominant trait. You would write it in lowercase letters. But this is not a complete dominance. It's incomplete. And therefore, you would write CW, CW. So you underline one from each pair. And so what do the gametes have? Capital CR, capital CW. Now, so each gamete would have CR, CW, male gamete, female gamete would also have CR, CW. So when you do the cross, what we see is when you cross CRCW with CRCW, so cross CRCW with CRCW, you get CRCR, CRCW, CRCW, don't put W first, and CWCW. What does that mean? CRCR, red snapdragon, CRCW, it's the heterozygote. So instead of getting red color, we got pink. Let's use the analogy. When you take red paint and you mix it with white paint, what do you get? Pink color, pink paint. And that's what Mendel saw. He saw a new trait, a new color, which was called pink color. So he says, oh, red is not definitely completely dominant over white because we see a new trait called pink color. And therefore, he said that red color is not completely dominant over white, but the heterozygote, which is pink color, is lying in between that of the dominant and the recessive trait. And so you get one homozygous dominant red with two heterozygotes. The heterozygote is now pink color with one completely recessive white flower. So you got red, white, and pink flower, red, white, and pink flowers. So this was the law of incomplete dominance. Now he put forth another law called law of codominance. What does law of codominance states? That two dominant alleles are going to affect the phenotype, the outward appearance, in separate ways. So we're going to see classic examples of codominance. I'm going to talk about genetic disorders. The first genetic disorder that we are going to touch upon is Tay-Sachs disease. Tay-Sachs disease is a very fatal, dysfunctional. It's a disease due to a dysfunctional enzyme. Dysfunctional enzyme means an enzyme that is not functioning correctly. And as a result, because this enzyme is not functioning correctly, there is an accumulation of lipids in the brain causing a lipid-brain barrier. Now, at the organismic level, what happens? The allele is recessive. At the biochemical level, the phenotype, that is the enzyme, which refers to the enzyme activity, is incompletely dominant. And at the molecular level, the alleles are codominant. So this is an important slide to take note of, these terms. What happens at the molecular level? What happens at the biochemical level? What happens at the organismic level with respect to the allele? And when we say fatal, meaning these individuals are not going to live, they die. So let's look at, I will talk about the genetic disorders a little later. Let's look at multiple alleles. What we mean by multiple alleles, analyze the term, many alleles. Now, we know that most of the genes that exist in populations are going to exist in more than two allelic forms. For example, we are looking at our blood types. We have 4 phenotypes for the ABO blood group, which is A, IA, IB phenotypes, AB, ABO. IA, IB would be the enzyme that refers to that particular allele. Okay, so when you look at the ABO blood groups that are determined by 3 alleles for that particular enzyme, I refers to the enzyme, and A and B are referring to the type of carbohydrates that are going to attach to that particular blood cell. And the enzyme is encoded by the IA allele, will add to the A carbohydrates. Enzyme encoded by the IB allele adds the B carbohydrates. And the enzyme encoded by that lowercase I will add neither A or B carbohydrates. So the alleles are IA, IB, lowercase I, meaning no carbohydrates is going to attach itself to that blood type. Carbohydrate A is triangular, carbohydrate B, they represent it as a circle, and lowercase I has no carbohydrate attaching. So if I asked you, what were the genotypes for the blood group? The genotype for A blood type, the phenotypes are A, AB, A, B, AB, O. Now, the genotype for blood type A is represented as IA, IA, or IA lowercase I. It may not have that carbohydrate attaching. This is homozygous condition, heterozygous. And clearly we can see A carbohydrate is attaching to the red blood cell. Blood type P individuals have B carbohydrate attaching to the RBC, red blood cell. So how do you write the genotype? IB, IB, or IB lowercase I? If you are blood type A, B, you have A and B carbohydrate attaching, so you would only write IA, IB. I am blood group O. Blood group O individuals are called the universal donors. So I have no carbohydrates attaching, so my genotype is lowercase I, lowercase I. But what is so important about blood type O individuals? Why are we called the universal donors? Because we can donate blood to A blood group, B blood group, AB, and O. But I can only receive blood from another O individual. So now let's analyze. Whom can an individual who has A blood type donate their blood to? They can donate it to an AB, to an A, and AB. Can they donate it to an O? No. Who can blood type B individuals donate to? They can donate to another blood type B, AB. Who can AB donate to? A individuals, B individuals, AB. But blood type O can donate their blood to an A blood group, B blood group, AB, and to themselves. But they can only receive from another O. Okay. So now we come to what is pleiotropy. When genes have many phenotypic effects, and the property is referred to as pleiotropy. For example, pleiotropic alleles are responsible for many symptoms, multiple symptoms of certain hereditary diseases such as CF, which is cystic fibrosis, and sickle cell disease. Now, in epistasis, you have to know these terms. In epistasis, the expression of a gene at one locus and the structure of a chromosome, again, I keep emphatically stating, in the structure of a chromosome, I gave all these terms. What is the locus? It's the location or the position of a gene on a chromosome. And therefore, in epistasis, the expression of a gene at one locus is going to alter the phenotypic expression of a gene at a second locus. So now we learn something called polygenic. Poly means many. What's polygenic inheritance? It is the additive effect. So when two or more genes determine on a single phenotype, determine a single phenotype, the effect, the additive effect of two or more genes has on a single phenotype is referred to as polygenic inheritance. Now, height of an individual is a very good polygenic inheritance. Normally students, every gene specifies a particular trait. In other words, every gene is going to determine our trait. Genes for hair color, eye color, blah, blah, blah. But for skin pigmentation, many genes are responsible for our skin pigmentation. Same way, polygenic inheritance. We have over 180 genes that are responsible for the height of an individual. Many genes, polygenic, multiple genes affecting. And as I told you, skin color. So this is all polygenic inheritance. Now we want to know, why don't we take humans to study or to conduct genetic experiments? Because humans, the generation time is too long. Parents are going to produce relatively fewer offspring, and the breeding experiments are unacceptable. It's against the law to conduct genetic experiments on humans. But Mendel studied the pea plant. The reason being, the pea plant, it's easily accessible, inexpensive. Pea plants had all these opposite contrasting traits, and mainly because both the male sex organ, which was the stamen, and the female sex organ called the pistil, was present in the same plant. So it was easy to self-pollinate those plants. So we're looking at how do these recessive alleles behave. Recessive alleles are only going to show up in individuals that are homozygous for that condition. Now, carriers, who are carriers? Carriers are always heterozygous individuals who are going to carry the recessive allele, but they are phenotypically normal. Now, most individuals with recessive disorders, they are born to carrier parents. Albinism, albinos, an individual is referred to as an albino, is a recessive condition which is characterized by lack of pigmentation of skin and hair color. So when you look at the albinos, you see she is an albino. She's not, because you see her eyelashes will be white, hair color, skin pigmentation, extremely white, lack of skin pigmentation, and therefore they are called albinos. So we see here heterozygous parents, the phenotype. When you mate a heterozygous with another normal phenotype, which is heterozygous, the normal phenotype will be capital A. The carrier is heterozygous, but the albino will be lowercase a. Okay. Now, CF, we talked about CF. What is cystic fibrosis? Cystic fibrosis is the most lethal genetic disease, which is very predominant amongst European descendants. It strikes 1 out of every 2,500 people of European descent. It results in a defective or absent chloride transport channel. So these individuals who suffer from cystic fibrosis, they have either they lack the chloride channels, transport channels in the plasma membrane that we learned when we studied the structure of plasma membrane, or they have a defective chloride transport channel. So chloride ions are not able to pass through, are not transported across the plasma membrane. And as a result, what would happen is it leads to a buildup of all chloride ions outside the cell. Therefore, what are the symptoms these individuals will suffer from? There will be a lot of mucus buildup in the internal organs and abnormal absorption of nutrients in the small intestine. And this is a lethal genetic disease, meaning these individuals will ultimately die. Let's look at sickle cell disease. Now, in sickle cell disease, sickle cell anemia, sickle cell disease is predominant amongst African Americans and Africans and their descendants. And 1 out of every 400 African Americans is affected by sickle cell anemia. Now, what is sickle cell anemia on account of? There is a substitution of a single amino acid called glutamic acid with valine, VAL and GLU. And I'm going to explain it out here. So it has to do with the blood cells, hemoglobin, the protein, the transport protein, the globular protein. Our normal red blood cells are round and they are flexible. That's important. Individuals who suffer from sickle cell anemia, their red blood cells are abnormal, and they are sickle shaped, crescent shaped or moon shaped. And they are stiff. So if you have capillaries, which are the thinnest blood vessels, what happens? Since I do not suffer from sickle cell anemia, my red blood cells are nice and round and flexible. So they can easily maneuver through the thinnest blood vessels called the capillaries, thereby supplying oxygen to the tissues, to the organs that they serve. But in individuals who do not have, who suffer from sickle cell anemia, who do not have normal red blood cells, their red blood cells are distorted, abnormally sickle shaped, and therefore, and because they are stiff, they start accumulating in the capillaries. Now, when they aggregate, accumulate, they all stuck together. So what happens? No oxygen can pass. There is deprivation of oxygen to the tissues, to the different organs and organ systems. And as a result, these individuals will die. And all this is on account of substitution of one amino acid, glutamic acid, with valine, VALIN. So, but these individuals, what are the symptoms? They go through excruciating pain, fever, weakness, organ damage. Very often they have to go through blood transfusion. And sometimes they even suffer from paralysis. Now, I want to say one thing that comes out of sickle cell anemia, and I like to use it as a positive part of sickle cell anemia, is individuals who suffer from sickle cell anemia, what happens to them is that they will not suffer from malaria. Now, why? Because their red blood cells are distorted, abnormal shaped, sickle shaped, crescent moon. So when the mosquito goes to attack it, remember they're stiff, so they cannot penetrate and enter and cause the infection. But our cells, red blood cells, which are normal, now flexible, the mosquito comes, attacks, and it can enter with its proboscis, infect us, and cause malaria. Where is malaria caused? In Africa. There is a lot of rainfall, marshy areas. That's a breeding ground for mosquitoes. And what does the mosquito carry along with it? The mosquito carries along with it a parasite called Plasmodium falciparum. I'm just giving it to you. You don't have to remember this, but it's nice, good knowledge. Excuse me, student. So, Plasmodium is Plasmodium. Falciparum, Falciparum. Plasmodium falciparum. And so when it injects it, that's how it's causing the malaria. Now, who is a carrier? A carrier is an individual who carries the allele but does not suffer from the symptom. So if the individual suffers from sickle cell anemia, they have to be homozygous for that condition. A carrier will be heterozygous. So normally, how do we denote sickle cell anemia? We would say H for hemoglobin, HS, capital HS, capital H, lowercase s, is homozygous for the allele. So that individual suffers from sickle cell anemia. A carrier would be capital HS, capital HA. And a normal individual will be capital HA, capital HA, you're homozygous. Okay. Let's go to another disease called Huntington's disease. Now, students, this disease is really, I say, we do not know whether you suffer from the disease as we can be afflicted by this disease and the symptoms do not show up until you're between the ages of 40 to 50 years. Some textbooks show 35 to 40, some show 40 to 50. Now, what does this Huntington's disease affect? It affects the nervous system. So it's a degenerative disease of the nervous system in which the individual shows loss of coordination of the muscles. So they cannot coordinate the hand and leg movement. And this disease is fatal. Let's go to what is fetal testing and amniocentesis. So fetal testing and amniocentesis, the liquid that bathes the fetus, is removed and tested. We have two techniques, amniocentesis and chorionic villus sampling. So I'm going to explain this. These two techniques are used to determine whether any genetic disorders exist. They can be determined at birth by these simple tests, which is routinely performed in hospitals in the United States. And one common test for PKU, a recessively inherited disorder, occurs 1 out of every 10,000 to 15,000 births in the United States. So let's look at this. In amniocentesis, what is amniocentesis? You may have heard of it. It's a technique in which the physician or the doctor will insert a sterile syringe into the amnion. Can you see this reddish brown area? Is the amnion, A-M-N-I-O-N. The amnion is a sac in which the fetus is developing. And the fluid is referred to as amniotic fluid. So the doctor is inserting through an ultrasound monitor the sterile syringe and withdraws some amniotic fluid. But what does this amniotic fluid contain? This amniotic fluid is going to contain some fetal cells. Now, when is this amniocentesis carried out? It's carried out between the 3rd to 4th month of pregnancy. These fetal cells are then grown and centrifuged. When centrifugation, it's spun at a high force, at a high speed, and some cells will collect at the bottom, what we call the pellet. These fetal cells in the pellet are then grown in a petri dish in the lab. After several hours, biochemical and genetic testing is conducted and results are obtained after several weeks, where a karyotyping is conducted. We had this karyotype under structure for chromosome again in mitosis. And so what happened was this is only got after several weeks the results. Let's go to CVS, chorionic villus sampling techniques. In this technique, this physician, surgeon, or doctor inserts a sterile tube into the amnion. Here it's smaller, into the chorion, pardon me. The chorion is this pink layer, this pink sac. So you can see the chorion here. However, these are the two embryonic membranes of the fetus, amnion, chorion. Chorion surrounds the amnion. So again, looking through an ultrasound monitor goes through the cervical region into the uterus, and they get a scraping of the chorionic villi tissue, which will contain some cells. These fetal cells are centrifuged, collected, and once again, biochemical and genetic testing is carried out. But after several hours, results can be obtained. So this technique is carried out as early as the 11th to 15th week of pregnancy. Why is the chorionic villus sampling technique much more advantageous? This is a tube. In a tube, you're going to get higher concentrations of cells, more concentration. In a syringe, fewer cells. So this is more advantageous, carried out as early in pregnancy. And thirdly, because after several hours, karyotyping can be carried out. You're not waiting for several weeks when the damage has been done. In the karyotype, they're looking to determine if there is any genetic disorder, abnormality, more chromosomes, less chromosomes, and also if there is any abnormality with the chromosome number and size. So we will be talking about now two sex disorders, sex-linked disorders, which is Turner syndrome, Klinefelter syndrome. So we normal humans will have 46 chromosomes, the 22 pairs, which are autosomes, and this chromosome. So they can identify the sex of the individual. If they see this is a Y chromosome, so this is a human male karyotype. If they have one missing chromosome, like XO for Turner syndrome, then they say the individual is Turner's. And so here I've just put in a chart at a glance that you can see the incomplete or complete dominance. So Turner syndrome and Down syndrome and Klinefelter's. So what happens in Klinefelter syndrome? In Klinefelter syndrome, what I'm going to just show you here is open a new PowerPoint for you so I can just write these words and explain the Klinefelter syndrome to you. Over here is in Klinefelter syndrome, you have, share the screen here. In Klinefelter syndrome, what we have is, I'm going to write on a whiteboard. So I don't have, I can just write Klinefelter syndrome. Klinefelter, F-E-L-T-E-R, Klinefelter syndrome, S-Y-N-D-R-O-M-E. And Klinefelter syndrome, I want you to write into parentheses X, X, Y. So what happens in Klinefelter syndrome? You have when an XX individual mates with a Y individual, the resulting zygote is XXY. So what happens here is, let me just write this properly for you. I can put here, the resulting zygote is XX and Y. Okay, that's the zygote. So the resulting zygote is XXY. So what happens to these individuals, XXY? These individuals, they are I'm in my office trying to write down, so just playing first time in my office screen. So what happens to this? Oh, I said it is, we don't want this. So what happens to these individuals which are XXY, right? So these individuals which are XXY, they have, right here, XXY. So these individuals who are XXY, they have XXY. This is the zygote. Remember, you have a total of now 47 chromosomes. These individuals have 47 instead of the normal 46 chromosomes. So what happens to a Klinefelter's individual? These individuals, they are sterile male. Now, because you see the Y chromosome is present, therefore they have to be males. So these individuals are sterile male, S-T-E-R-I-L-E, males. But what do they show? They have enlarged breasts and the pubic, they have pubic hair in the male sex organ. They have high pitched voices, P-I-T-C-H-E-D, and they have smaller male sex organs, which is the penis. So of course, these males, they can ejaculate sperm, but their sperm is not fertile, so they cannot produce children. Now, if you look at the other disorder, which we call Turner syndrome, T-U-R-N-E-R, Turner syndrome. An individual with Turner syndrome, into parentheses, you're going to put XO. What does O stand for? O stands for the missing chromosome. So these individuals are going to have 45 chromosomes rather than the 46 chromosomes. Now, what is that missing chromosome? It's an X chromosome. So once again, these individuals, they have underdeveloped sex organs. They will have so therefore they cannot produce eggs. They're supposed to be females. And they do not have the total 46. And so when they go through the menstrual cycle, they don't go through proper menstruation. That's why they don't ovulate. And so that's their genetic disorder. And the last genetic disorder is Down syndrome. So if I talk, we are familiar with Down syndrome. Now, Down syndrome, these individuals, they have 47 chromosomes. Now, this is not a sex-linked disorder. This is a autosomal linked disorder. Autosomal meaning it is linked to the 22 pairs of chromosomes, autosomes, autosomal linked disorder. What happens to these individuals in Down syndrome? Where is this chromosome, extra chromosome? This extra chromosome is on chromosome number 21. So instead of having a pair of chromosomes, these individuals will have 3 chromosomes, an extra copy on chromosome number 21. So they end up with 47 chromosomes. Now, why do they have this extra chromosome? So because they have 3 chromosomes, the condition is referred to as trisomy, Trisomy. Trisomy is the condition. And so trisomy is the condition. And what is this due to? It is due to a process what we call non-disjunction. Very important term. You will be asked these in the exam. So I can also go through this in the lab with you so that we can better understand, because I'm just writing it on my screen here from the office after your lab today. So what is non-disjunction? Non-disjunction is the failure of this extra copy of chromosome to separate out from its pair. Now, where this extra copy came, geneticists don't know. And because it failed to separate out from the pair, it has caused this aberration called Down syndrome. Now, what are the symptoms? These individuals, they have folded skin, loose folded skin around their eyes, which tend to slope up at the corners of their eyes. Then, many of them, they are mentally retarded. Their IQ is lower than average. They won't go through proper menstrual cycle. But in very often, you could also have it on chromosome number 15, where the intelligence level is much higher. Then the back of the head is flat. Then we see the speech is faulty. These are some of the symptoms of Down syndrome. So we can see now how Down syndrome has affected so many individuals. And the highest rate of incidence of Down syndrome is in the US. So I hope you enjoyed this lecture, students. Have a great one. Bye-bye.